Welcome to this biochemistry lecture on biomolecules, where we'll discuss the four biopolymers: nucleic acids, proteins, lipids, and carbohydrates, as well as hormones. So biomolecules compose most elements of a cell and an organism. They are commonly a hydrocarbon backbone that's ornamented with different functional groups to generate some kind of structural element or motif, and that structure then is important in its general function. So what we find is structure equals function, and that's true for all these different biomolecules we're going to talk about today. There are four major classes of biopolymers: carbohydrates that are composed of monosaccharides. So those monosaccharides are linked together to form a long chain polysaccharide that are important in many biological functions. Lipids, where fatty acids are linked together on a polar head group. That fatty acid, that lipid, is then interacts with other lipids, and a very important structural function in things like membranes. Proteins are long chain amino acids that are important, and they form a three-dimensional structure and perform a lot of the work of the cell. And nucleic acids, which are the informational storage unit for cells. We take nucleotides and link them with a phosphodiester into long, very long chain structures that again store information. Again, all of these biopolymers are hydrocarbon backbones that are ornamented with functional groups to generate a structure that drives their function. The important reaction is a dehydration synthesis reaction or a condensation reaction, and commonly what we find is a short polymer chain. We add a monomer to it through this dehydration synthesis reaction in order to keep getting longer and longer polymer chains. Again, a dehydration reaction, a dehydration synthesis reaction, or a condensation reaction drives all of these biopolymers into making them very, very long structures. The first example is a carbohydrate, where we take monosaccharides, commonly a ring, for instance glucose, or 5 or 6 carbon monosaccharides that are in a ring structure, and we take and they're linked together with glycosidic bonds. So there's a glycosidic subunit that puts together, there's a condensation reaction that occurs that generates that glycosidic bond, and we just keep adding these on, and sometimes they're branched even. They can get to be really complex. Polysaccharides provide an important energy storage, an important structural component in things like cellulose and chitin, and they're also an important element in the DNA and RNA subunits. Lipids are formed when we have a polar head group that we take and we form an ether linkage, but it's a condensation reaction with fatty acid, long chain fatty acid, and these long chain fatty acids are aggregated into membranes due to their hydrophobic effect, and that provides an important element, structural element within a cell, where we protect the machinery of a cell, and we provide an electrochemical gradient that provides metabolism. So these lipids perform important functional based on their structural elements. Proteins are amino acids that are linked together in a peptide bond. Again, a peptide bond is a condensation reaction that will allow these proteins to generate molecules that are several hundred, if not thousands, of amino acids long. Those provide important working elements within the cell, including things like enzymatic activity, biosignaling, structural elements, and things like keratin and collagen. And they also provide transportation mechanisms. So all of these biopolymers have important structural elements that then lead to their function. In particular, proteins are the quintessential example of this because of their diversity of functionality that they have. Nucleic acids are nucleotides that are linked together with a phosphodiester bond, which is in this diagram is shown in purple, and there is an element where the structures link hydrogen bond together, two DNA strands hydrogen bond together, but the main structural element of this is the phosphodiester linkage that links the different nucleotides together. That provides the information. And so a nucleotide, which is composed of a nitrogenous base, the ribose, a ribose sugar of some kind, a deoxyribose or a ribose, which is in this example is shown in red, and a phosphate linkage, which in this case is shown as in purple, that provides the structural element and the compacting nature of the information of this the DNA, and thus the information inside this cell. Hormones, again, they're the only non-biopolymer here. They're a long chain, sometimes very complex, as you can see from all the ring structures, hydrocarbon with functional groups, and sometimes the functional groups aren't numerous, but their location is very important, as is the case in this this example that we're showing here, where we have two oxygens, one's a hydroxyl and one's a carbonyl. Both of those are important structural elements within a hormone, which are commonly used for signaling in our cells. Again, biomolecules commonly have several functional groups that provide important functions within that cell, and they can be as complex as or simple as a carrier. In this situation, this is acetyl-CoA. Acetyl-CoA is a carrier of the acetyl functional groups, and the different functional groups that ornament this hydrocarbon backbone provide functionality, allow it to interact with different molecules in specific ways, and that specificity allows important reactions to be regulated and controlled. And so really, when we're talking about these biomolecules, structure equals function, and function is dictated by the location and type of functional groups we have, and that provides specificity and tight control of all of the biochemistry that's associated with a cell and also an organism. With that, we'll end this lecture, and we look forward to discussing some of the other topics that are surrounding this, including functional groups and some of the other bonds that help build up these biopolymers.